Hi, I'm Paul McElhany with the NOAA Northwest Fisheries Science Center. And what I'm going to talk about today is some recent experiments that we've done looking at the potential effects of ocean acidification on Dungeness crab. At the very end, when we're looking forward, I'll talk about some projects that we have going on other species. So the work that we're doing is responds to several themes in the 2010 NOAA Ocean Acidification Research Plan, particularly Theme 2, looking at the responses of marine organisms. And in Theme 3, we've done other work that I'm not presenting today, kind of focusing on predicting ecological responses to ocean acidification. So quick to begin with, a little Dungeness crab 101. Dungeness crab mate in the spring. The female then extrudes her eggs in the fall, nestles down into the sediment in the spring, kind of January, February time. The eggs hatch into planktonic zoeal stages, go through 5 larval zoea stages that then molt to the megalopy stage, which is sort of a transition stage to the juveniles, which then settle down onto the seafloor. And then another 2 years, they're able to mate and sort of start the whole cycle all over again. Also, a little bit of background, it's kind of a current snapshot of Dungeness crab populations. They're stable, generally coast-wide, or perhaps slightly increasing, with a few notable exceptions. Here in Puget Sound and in the South Hood Canal, some of the populations are declining such that they're no longer able to be harvested. Trying to understand what's going on there, kind of leading potential hypothesis as temperature, but as we'll see from our work, ocean acidification could play some role. So to quickly describe the experiments that we did last year, looking at Dungeness crab zoea, we collected gravid females from out in the Strait of Juan de Fuca, brought them back into the lab. When those zoea hatched, we placed them into these CO2 chambers, which are chambers where we equilibrate water that we put into the well plates where we're rearing the crabs. And then the atmosphere inside of the chamber is also made at our target CO2 conditions. So we simulate current CO2 conditions and then future ocean CO2 conditions. Here on the right, you can see a little zoea swimming around in one of these well plates. And this little kind of ghost zoea there in the right is the molt of that zoea. So we can track sort of their survival over time, and we can look at their developmental rate as they move through these different zoeal stages. Doing this is very labor intensive. It takes we need to look at a lot of different zoea, and so there's a lot of, we check these every day to follow survival and molting, and so there's a lot of looking involved. Here's a summary results from that 2019 experiment. And what we see is that we have, on this axis is time since hatching, and on the y-axis is the probability of survival. And what we see is that there's a higher survival in the lower CO2 treatment than we have in the higher CO2 treatment. Looking at developmental rate, here again we have on the x-axis we have the time since hatching. Each of these kind of paired colors, the solid is the low CO2 treatment, and the dashed is the high CO2 treatment. And so what we see is that there's this slower development rate of the time in which they transition from the Z1 to the Z2 stage, or the Z2 to the Z3 stage is slower under high CO2. Those were just sort of summary snapshots. When we look at variability, this is an experiment that we did this year. We just kind of managed to squeak in pulling out these data before we shut down the lab for COVID. And what we see is that we have this variation among parents within a population and among different populations. In this study, we collected crabs from Puget Sound proper, from Hood Canal, and then from the outer Washington coast. And then we see, like within Puget Sound, in some of the populations, there was an effect of CO2 from their offspring, and for others, not so much. And then when we compare across populations, the outer coast, we didn't see really any sort of statistical difference from the effect of the CO2 treatment, whereas we did for at least some of the parents in the other populations. And we're expanding this study, or plan to, this next year by looking at some populations up in British Columbia in a collaboration with DFO Canada. Looking at a different life stage, we did an experiment in 2018-2019 looking at the Mukilteo stage, which we see sort of scampering up across this tube right here. Some of the smaller pieces that you see across there are some zoea that are skittering across, and the little bits are some Martinia that we feed to the crab. What we did was we collected Mukilteo from light traps out in Puget Sound, brought those into the lab, and placed them into different CO2 treatment conditions. The Mukilteo quickly, within a few days, generally molted into the juvenile stage, which you see here in the bottom right. We reared them in these high and low CO2 treatments for almost a year, and followed again their development because we could track the molting. We looked at them every day, as well as their survival. In this case, sort of contrary to what we see for the zoea stage, if anything, it appears that survival is higher in the high CO2 than for the low CO2 conditions. Likewise, we don't see any kind of effect of the CO2 on developmental rate. We've looked at size as well as metabolic rate, and there are sublethal, kind of effects that we see on size of the juvenile crabs and on their metabolic rate, but nothing at this kind of gross scale of survival or development. These stages seem fairly hardy, and as you can see, the pH was quite low in these treatments, down to 7.15, which is low even for Puget Sound. So some sort of takeaways from these experiments is that the Dungeness crab populations are generally okay, so we don't see any kind of big effects of either CO2 or climate change on the populations coast-wide. We do see a higher mortality in zoea, but no effects, or if anything, a higher survival under high CO2 for the Mukilteo and juvenile stages, but with some sublethal effects. And one other kind of observation is that attributing OA effects to Dungeness crab in the field is very challenging, because there are a lot of things going on. We're still trying to, and kind of a long-term focus of our research is trying to understand the impact of OA on Dungeness crab. And the reason why we're focusing on Dungeness crab is because of the most valuable fishery on the West Coast in terms of sort of cash value. And so that, combined with sort of our previous work suggesting that they're susceptible to effects of high CO2, has led them to a kind of a major research focus for the lab. So kind of looking forward, these are kind of just some snapshot images from our next work plan, so our 21-23 work plan with the Ocean Acidification Program, and these are projects that will be funded through the OAP. I want to continue the crab work, trying to get at the mechanism for the survival differences that we see, and to try and understand observations about potential changes in exoskeleton condition correlated with field conditions, some work by Nina Bidnarsek. And to do that, we're using stable calcium isotopes to label the crab carapace so we can follow what happens through the molting cycle. Another project that's led by Andy Dittman here in the upper right is looking at sort of neurobehavioral effects of CO2 on salmon and sablefish. This is work that he did the sort of the last cycle and showed that under high CO2 that there are behavioral effects, and they're trying to understand the mechanism by what's going on by looking kind of at the activity in the brain. This is a clarified salmon brain, for those of you who didn't immediately recognize it. I know it seems pretty obvious, but they are able to kind of identify exactly which regions of the brain and which neurons are affected under high CO2 under these olfactory stimulation conditions. In the bottom left here, Shallon Bush is leading a project looking at the effect of krill, which are really critical in the West Coast food web. This is a collaborative project with Eric Yorkstead at the Southwest Fisheries Science Center, who's leading some modeling. So this is going to be coupled between experimental work that Challenge leading and some modeling work that Eric's leading to try and understand how effects on juvenile stage krill might affect happenings in the food web on the West Coast. And this final project is work on developing kind of an advanced acidification exposure system for a new lab. The Northwest Science Center is in the process of developing a new research facility up at Mukilteo that's explicitly kind of designed to include OA research. And we're developing a system kind of tailored to that new lab. And so we'll be prototyping that over the next couple of years as the lab is built. That's kind of all I had now, and so I'll stop. Thank you.